So thank you very much for that very kind introduction, and it's very nice to see everybody today. I want to thank the organizers of this conference for very kindly inviting us to speak and really tell you about our approach to cancer research, which we think is a little bit different. So just to give you some background, as many of you may know, we're actually approaching the 50th anniversary of the National Cancer Act, which was signed by President Nixon, back in 1971. And basically, as part of this act, there was a creation of a number of NCI-designated cancer centers. And the mission of these centers was really going to be to how do we take sort of the Apollo-like approach that led to reaching the moon and sort of rethink this as a way to how can we conquer cancer in the short time. Now, it turns out conquering cancer turned out to be a bit harder than getting to the moon because now 50 years on, I think there's a lot of discussion we can have about what kind of fantastic progress we've seen in cancer research over those 50 years, but clearly there's more work to do. But anyway, cancer centers evolved in two ways. There's the Comprehensive Cancer Center, the Clinical Cancer Center, which many of you may be familiar with. What separates those from the Basic Cancer Center, which is what we have here at MIT, is that a clinical cancer center, of course, has patients. The mission of the basic cancer centers is really to try to set the foundation, the knowledge base that will lead to the new therapies that can then be tested at the clinical cancer centers and hopefully ultimately improve the care of patients. Well, this led us to establish, or actually not me, but Salvador Luria, to establish the MIT Center for Cancer Research, which opened back in 1974. It was in a former candy factory across the street. And really, work at this center really was transformative in our basic understanding of cancer biology. In 2011, my predecessor as director of the Institute, Tyler Jacks, had a new vision that together with Susan Hockfield, I think changed the way that we certainly at MIT approach cancer research, but I think it's having far-reaching effects on how other places are also beginning to think about cancer. And their vision is that rather than have siloed scientists and clinicians who are working on very small problems or working on specific things, let's bring in a multidisciplinary approach where we take the best of MIT engineering as well as the best of MIT science, co-house them together, and see what happens. This, along with the transformative gift, led to the foundation of the Koch Institute, which led to the construction of a new building, which is our home, and continues to be what I think is sort of the center of cancer research, not just at MIT, but hopefully within Kendall Square and maybe even beyond. So we are housed in this building here. If you want to see our building, you can walk out the Kendall Square Marriott, turn right, and it's basically kitty corner just down the street. We have a lot of space. We are really sort of centrally located, very close to other famous MIT research centers such as the Whitehead Institute and the Broad Institute, as well as the fantastic neighborhood of Kendall Square and the Boston Technology Center. Now this slide here is actually a bit old, so if you're looking for your company or your favorite company up there and you don't see it, this is from 2017. I really need to take the time to try to update this, but you can imagine it's a lot of work to actually go through and try to keep track of the explosion of companies and biotechs that have really surrounded the MIT campus. But I think a real part of our innovation really derives from being part of this incredible ecosystem that we have here in Kendall Square. Now, our mission at the Koch Institute is to develop new insights into cancer, as well as develop new tools and technologies that will help us better detect, monitor, and treat the disease. So much of cancer research, we really think about the treatment piece, but of course, if we can find cancer earlier or before it even starts, that's going to have a much larger impact of anything we can do with treatment. And for those of you who are in the biotech space, you certainly know that the successful development of new therapies means we need to be able to monitor response. We need to know if those therapies are actually hitting the right targets and having the effect, because you can't tell success or failure if you don't really understand if your therapeutic hypothesis is working. Now, the real core to our center is our people. We are very lucky to have a number of intramural and extramural members of our cancer center. Extramural members are really anyone on the MIT campus who's interested in cancer research. But the heart of our center are really these people here. These are the intramural faculty of the Koch Institute, and I feel very privileged to come to work every day with some really amazing scientists and engineers who are listed up here, who I think are doing some of the most transformative and exciting work in basic cancer biology, translational cancer research, cancer-based engineering to really help us accomplish our mission. Now, we have constructed our space, that means the physical space of the Koch Institute as it was reimagined and rebuilt back in 2011, I think really has tried to be built in a way that helps us accomplish our mission. Now, there was an interesting challenge about bringing people together with different approaches, different ideas about how to approach cancer science and translational medicine. Engineers think differently than biologists. I don't think I have to tell people out there. And the cultures are somewhat different. What goes into design of an engineering lab, what was required for success, is a bit different than what's in a biology lab. But nonetheless, we wanted people to flow seamlessly and not really view themselves as scientists or engineers. And so the way the building was designed is we basically, on most of the research floors, it's basically set up as follows. You have an engineering side of the building and a biology side of the building. This isn't meant to silo people, but just to accomplish the practicality of having to have actually people work in their different places. And so, but central to that, we then have a great deal of interactive space that basically means that when people are not doing work, they come into this interactive space. And the vision is that if you work in a biology lab, you run into an engineer, and ultimately you don't think of yourself as a biologist or an engineer, but rather a Koch researcher who's really just interested in solving cancer. And I'm proud to say that that's exactly what happened. Now, also in the building, we have a number of spaces dedicated to various core facilities. The nature of science today is that it's very difficult to do all of the cutting-edge technologies that are necessary and house them within individual labs. And so we've built various individual cores that are located either on the garden level or on the first floor, a couple are on the second floor, where researchers throughout the building, and indeed throughout the MIT campus, and if capacity allows, even the entire Kendall Square area, can come in and access these core facilities where they really get access to cutting-edge equipment and the ability to use the best new technology to support their various research questions. Now, we have several specific core facilities. Some of these are things that are necessary just for the functioning of labs, things like glassware washing and media preparation, or the ability to house animals properly. However, we also have very cutting-edge techniques, things like high-throughput sciences, where our researchers can access the latest in high-throughput screening technology and robots in order to pursue whatever problems. Integrated genomics and bioinformatics, we can take advantage of single-cell sequencing. In fact, some of the single-cell sequencing approaches were actually developed by scientists within the Koch Institute. And I think a really exciting one related to our engineering mission has to do with our nanotechnology material characterization core, where our engineers in our building really can characterize all of the exciting devices or drug delivery tools that they are developing. Now, we center our research around 5 main focus areas. As I stated earlier, detection and monitoring. If we can find cancer earlier or better monitor its progression, that helps all stages of helping patients with cancer. If we can find it earlier, patients are going to do better. And if we can monitor response, that also is going to help development of therapies of all type. Metastasis is something patients die when they have metastatic disease. And so better understanding why patients have metastatic disease, how we can deal with metastatic disease, really central to the future of changing the reality of cancer for patients today. Precision medicine, something many centers are doing. The idea, how do we select the right patients? How do we match the right drug with the right patients? How do we develop the right drug with the right patients? Nano-based drug delivery. I often argue in my class to undergraduates when I teach cancer biology that some of the biggest advances in cancer medicine over the last 50 years isn't necessarily new drugs, but it's actually developing the ways to deliver those drugs, because if you can't get to the drug to the tumor, if you can't give the supportive care that the drugs can be given safely and effectively, patients aren't going to see the drugs and then they don't respond to them. And we have fantastic drug delivery research at the Koch Institute, and this is a very important area of focus for us. And our final area is relatively new, and that's immune oncology. You know, when I went through my training, I won't tell you how long ago, you know, people laughed at the cancer immunologists. Now, it's hard to be interested in cancer medicine and not focus on immunology. And of course, we have fantastic immune oncology at our place with a real strength in vaccines, but also better understanding various checkpoint therapies. Now, how we actually support research at the Koch Institute, of course, we rely on traditional government funding mechanisms, and we compete very well for those things. But we also realize that those things are not always supportive of true innovation, and so we need a number of programs that help us deal with innovation, as well as at the same time move in a direction that links us closer to the clinic. And we have a number of initiatives here. We've built centers within the Institute that allow us to have subgroups of people where they can access resources and deploy those in a direction to lead to new projects. But one that I want to talk a bit about today is the Bridge project, and I want to talk about that simply because this is I think a transformative program for us that really has allowed us to take the innovation at MIT and move it much closer to the clinic. What this is an entirely philanthropically funded effort that is a collaboration between our center at MIT and the Harvard hospitals, part of the other NCI-designated cancer center in town, the Dana-Farber Harvard Cancer Center, which really encompasses the cancer center of the various Harvard hospitals. And the way this works is that MIT scientists have to partner with someone in the Harvard hospitals. They co-develop projects together. These should be things that are either near-term going to make a difference in the clinic, or perhaps might even be early-stage clinical trials. These are funded, and I think they've led to some real innovation. And in the remainder of my time, I want to take you through a few vignettes to highlight some of the exciting things that are going on at the Koch Institute. I'll start with something that was initially funded through the Bridge project and is now moving into the clinic, also leading to new company creation. And along the way, I think you'll get a sense of how this idea of bringing together scientists, engineers, technologists, basic biologists, co-housing them together, having people work towards a common goal, really, I think, is leading to exciting new innovative ways to tackle the problem of cancer. And so this first problem I want to talk about was a very cool device that was developed by my colleague Scott Manalis. Now, Scott Manalis is a bioengineer. He's really decided to use the biophysical properties of cells as a way to, well, first of all, he came up with a very exciting idea to measure basically at femtogram precision the mass of cells. And he did this effectively developing this device called the suspended microchannel resonator. And the way this device works is that he uses microfluidics that single cells or single groups of cells can be flowed through this cantilever, and then the resonant frequency of the cantilever can then be related back to various physical properties of the cell, such as the buoyant mass and with other ways, the actual dry mass. He used this to do some basic science. In fact, we worked with him quite closely. My lab is interested in how cells grow, and so we could use this to really understand some really cool things about the biology of cell growth. But as a way to translate this to clinical scale, begin to make a difference in translational cancer medicine, they then engineered a way to do this simultaneously across many, many different cells, which then gives you the way of using this as a biomarker or a pharmacodynamic assay, if you will, for actually what might happen in patients. He coupled together with scientists through the Bridge Project, with clinicians through the Bridge Project who are interested in multiple myeloma. This is a disease of malignant plasma cells. And he basically went and correlated the biophysical properties of how treatment of those plasma cells with various drugs actually led to changes in biophysical properties, and then they could go back and say, how did that relate to what actually went on in the clinic? This led to this very virtuous cycle where they could treat patients, isolate samples at the same time, study the response in the microchannel resonator to the drugs given to patients, compare what they see to the clinical response, and then are using that to actually drive decisions about whether or not the patient should be switched to a new drug. This is leading to very exciting clinical trials, and it's also led to the foundation of a company, Travera, which Scott Manalis founded as a way to try to go forward and and expand this larger to the clinic. Another exciting technology that was developed by my colleague Michael Sema, also initially through funding from the Bridge Project, which is also funding the expansion of this into clinical trials, has to do with, we know that oxygen can be very limiting for radiation therapy. It's been known for decades that the way radiation therapy kills cancer cells requires the presence of oxygen and the generation of reactive oxygen species. And this means that radiation dose depends on what the oxygen level is present. However, it's very difficult to assess radiation dose at the actual point where you're delivering the radiation. And so he came up with a device that's very small, can be implanted, and actually in real time measures out what is the oxygen concentration within that tissue. He applied this to cervical cancer, where basically you can implant this device together with brachytherapy, which is near-dose radiation delivery, and basically use real response based on oxygen level to time what is the radiation dose that should be delivered at this site. This is now also in trials, and I am quite excited about this as something that could make a big difference in the treatment of these patients with cervical cancer. The next thing I want to talk about is work of my colleague Sangeeta Bhatia. So Sangeeta has long had an interest in developing nanotechnology-based sensors to monitor and detect disease. And effectively what Sangeeta did is built an entire library of protease-driven sensors. Effectively, this is a nanoparticle where there is a probe on it that when cleaved with a protease leads to release of a piece of the cleaved piece off, that can then be detected in various ways. Often in the lab, we use fluorescence-based detection, but this can be changed in other ways that, say, could be detected on a paper strip. One of their lead programs is they're using this for lung cancer detection, where the patient will inhale this nanoparticle, and then they read out the presence of the cleaved parts in the urine as telling you something about whether or not this patient may have an early lung lesion. And this is actually looking quite exciting. And in fact, this technology, what's nice about it is it's scalable in other ways. Many cancers have altered protease activity. One can take advantage of altered protease activity in many, many different ways and use this as a way to monitor many cancers, we hope, but in addition, perhaps also even non-cancer-based diseases. And Sangeeta, entrepreneurial that she is, also started a company around this technology called Glimpse Bio, which I think is doing quite well. Another example that I want to share with you is work that was done by my colleague Richard Hines. This is really a story that starts from basic cancer discovery. The Hines lab, Richard's lab, has been interested in a long time about the extracellular matrix. This is the non-cellular components of tumors. Many, many years ago, they started a project to just go out and characterize what is the extracellular matrix of different cancers. That is not what cells are there, but what are the proteins that are surrounding those cells within the tumor. He termed this the matrisome and is an omics that you don't often hear discussed. But in doing this, by going through and developing the matrisome across different cancers, they found that they were unique extracellular matrix signatures that had to do with specific cancers. In particular, pancreatic cancer is one that they have focused on a lot, something that's very hard to detect early, hard to monitor because of its location within the body. Based on this, they then developed single-chain monoclonal antibodies, so so-called nanobodies, that can then target specific things about the extracellular matrix in pancreatic cancer. And as shown here, these can actually detect within, at least mouse models, very nicely where the cancer is located. You can see the cancer there as it comes around. They're picking up the pancreas cancer. You can see some of the probe excreted in the kidneys and the bladder as well. And they're quite excited about this as a way to drive earlier detection of pancreas cancer, as well as monitor the disease. A very exciting technology that was developed by Oliver Jonas when he was a fellow working with Bob Langer and Michael Sema, that is also something that has moved on to the clinic, is a microdevice that allows one to assess drug response in real patients and help guide therapy. It's been known for a very long time that screening cells in culture doesn't always reflect what happens in the body. And of course, we care if our drugs are actually going to work in the body, but it's very difficult to test multiple drugs, multiple combinations in the same patient. Well, what Ollie came up with was a device where he has a number of microwells on it that he can load with different drugs or drug combinations. He can deliver this into a tumor on a biopsy needle, leave it in the patient. The patient then goes to surgery or has that device retrieved with a second biopsy needle. And then they can actually look at, in the tumor by histology, registering to each place on the microdevice whether or not there was a response seen within the tumor. This is now moving forward to be tested to say, can you actually predict responses in patients, as patients will have this done and then go on to receive standard of care. We know some patients will respond, some patients don't. It'll be very exciting if we can predict who's going to respond or not to existing drugs, standard of care, but of course the real excitement here is for those who won't respond to existing care, can we then predict what is the right new combination that that patient should receive? What's great about this and really illustrates the excitement of the Koch Institute is some work that we did with Ollie that was like, we recognized that this could be used to also answer scientific questions. And we had discovered with researchers other places that a process called macropinocytosis, uptake of bulk extracellular material, could be an activity that is happening in pancreas cancer. But we had no idea if this was happening in vivo because we don't have the way to deliver the stuff to actually look at this. We realized we could actually repurpose Ollie's device in our mouse models to actually ask a scientific question about how much protein catabolism was actually happening within pancreatic cancer tumors. We used this and found very excitingly that indeed this is going on and launched an entirely new direction of research for us as well as others in the field. The very last example I want to give you is something that I'm quite excited about, which is sort of how we can apply the exciting advances in computation, machine learning to changing healthcare. This is, of course, something on many, many people's minds, and this is the work of my colleague Regina Barzilay, which really led to some work she did to help detection of breast cancer. Now, many of you know that women get mammograms, sometimes age in their 40s, sometimes in their 50s. There's a lot of controversy around that. And part of the controversy around that is that it turns out that younger women in breast density make it much harder to read the mammogram, but of course those are the people where it's much more important to find the earlier cancer. And in fact, breast density is an independent predictor of breast cancer risk. It's a difficult thing to read out. And in fact, there's a federal law that says that mammography providers actually have to tell patients about their breast density because it matters for their breast cancer risk as well as the ability to read a mammogram. Now, the problem with this is that if one just looks and takes a bunch of radiologists and says, all right, here's a bunch of mammograms, you tell me whose breasts are dense and whose are not, the concordance is horrible. So from one study, one group read 6%, another group read 84%, all right? That's almost useless in terms of actually using this in a clinically actionable way. What Regina realized is that one could use machine learning algorithms applied to the image analysis to try to address this problem. And so what she did is she applied her machine learning techniques to 40,000 mammograms that she got from her collaborator, Connie Lerman at MGH, and basically used it to assess breast density. And basically, what she found is that the algorithm she developed was 95% in agreement with who would be considered the most experienced radiologists. This has now been implicated into real clinical practice at MGH, and so this here is the pad that the radiologists use to sign out their report. There's actually an automated button reading there that uses Regina's algorithms. And what's even more exciting is now that you get this problem down, one can actually go on and actually address what one cares about most, and that is how can we better identify on a mammogram who's going to get cancer or not. Here's a mammogram of two different mammograms. It turns out one of these patients is going to develop breast cancer in 5 years and the other is not. I don't know how to read mammograms. I'm not a radiologist. I anticipate most of you aren't either. But a radiologist would tell you that there's nothing here that they would read out. And so Regina is now using these algorithms to really predict who is going to get breast cancer in the future. And I think we've now seen this type of approach copied everywhere, from machine learning to better read pathology slides, all the way to different imaging technologies. Hopefully I've illustrated for you that we're very entrepreneurial here at the Koch Institute. We definitely recognize that we are good at some things, but translation requires working with industrial partners, founding new companies. MIT is very forward-looking about founding new companies. This is through 2020. All of the KI-founded biotechnology companies, you can see it's rising every single year. And we are quite excited to work with you to share more about our science, and so please come see us if you want to. My colleague, Jane Wilkinson, our executive director, is here at this meeting. She's sitting here in the front row. She'll be around for the rest of the day. Come talk to me if you can grab me, but come talk to me or Jane, and we'd love to talk to you more about science at the Koch. Thank you very much. Great. All right, so I'm going to ask the question here. It's known that the patient's origin influences medical treatment and diagnosis. How do patients' geographic origin play a race in cancer research at the Koch Institute? That is a fantastic question, and it's actually one where I'm quite excited the future of machine learning could actually be applied. We know that there's only a certain amount of genetics that actually contributes to cancer risk. This has been looked at in numerous studies across GWAS and other things. We know that mutations alone are not enough to get cancer. Certainly cancer is caused by mutations, but it's been looked at. If one simply does the same qPCR test that looks for, that monitors BCR-ABL for CML, one finds that many people actually carry BCR-ABL positive DNA in their blood, but never get CML. So clearly other things contribute to cancer. You need the mutation, but there's other things that contribute as well. That's a hard problem to think about, but it's generally chalked up to environment. It's hard to think about environment, but I think this is a place where geographic origin and race play a role. Now, obviously race carries with it germline genetics, and polygenetic risks will be different based on race, okay? But I think people have looked at that, people continue to look at that, but a future direction that could be looked at is how does living in a different place, which begins to address some of the disparities in healthcare that we know are out there. We are in active discussions with cancer centers elsewhere in the US that have patients that have a range of served populations, and I think that this is really an exciting area where maybe we can use machine learning and other computational approaches to better understand the science of environmentally why there are different rates of disease in different communities. Am I out of time, or are there other questions? There was a second question, but it disappeared. Oh, there we go. All right. How long does it take to commercialize the technology that we introduce to consumers? That's an excellent question, and of course there's no one answer to that question, right? how long it takes to commercialize the technology depends on the technology and how quickly one can develop the various barriers that are necessary to actually commercialize it. In the case of Scott Manalis, this has gone on to, 15 or so years since he initially, well, I guess it also depends on when you say what's the technology. I mean, the idea for the microchannel resonator really is the history of his career and how that's evolved over the course of his career. From the time I think he realized that maybe this could be used in a translational way, few years to form a company, but then to actually see that to market, it's not to market yet, so the jury is still out on that. I think there's other examples, though. There's an example of a drug that I was involved with developing that back with Agios Pharmaceuticals, where, IDH mutations were discovered in 2007, maybe 2006, if you count when they were actually discovered and not published. And, drugs were in the clinic and approved around 2013, I believe, if I'm remembering properly. So that was quite a rapid time frame from discovery to implementation. There was some luck involved in that. Much of that was done in industry, so it wasn't necessarily the basic discovery that led all of that. But so I think really one has to look at the range and look at the specifics of the technology to really answer this question. What I can tell you is that our mission at the Koch Institute, and I think I can speak for MIT in this regard, is that we want to see technology translated, and we know patients need help. So we should translate it as quickly as we can. And lots of things go into how quickly things can be commercialized, from not just the science and where those barriers are, but funding and things like that, right? Sometimes you can go faster if you have more money. Sometimes you can't go faster because there's still something left to learn. And I hope going forward that we at the Koch Institute can tackle all parts of those problems. Maybe not the financial part, but at least the scientific and biological parts. Thank you.